Hi everyone. I decided it might help you to finish your lab report if I talked a little bit more about diffusion and osmosis. So let's start first with a definition of diffusion, which is the movement of molecules from a high concentration of those molecules to a low concentration. So you'll see in these diagrams, there's a high concentration of molecules over here, and eventually they keep moving through the membrane until they've reached an equilibrium, which means there's the same amount of molecules on both sides of the membrane. Now, what we worked with in our lab was actually a semi-permeable membrane, which means some molecules can fit through, but some molecules are just too big to fit through. So our plasma membrane in all of the cells in our bodies are a semi-permeable membrane, and so some molecules will fit through and some will not be allowed to fit through. Now, the fake cell that we made is also a semi-permeable membrane, and that's really because it has little tiny holes in it, and some molecules are small enough to fit through and some are not, which we found when we did our lab. All right, now I found a little animation of some molecules diffusing through a semi-permeable membrane. Now, in this video that I found, you're seeing some diffusion, and they say that it's here's the urine and here's your blood vessels, and here's the semi-permeable membrane. But we can look at this just like your artificial cell, remember, and you filled it with glucose and starch, and outside of the cell in your cup or the beaker was water, and you put iodine in it. Now, what I want to ask you, and take a look at this, is after you look at this for a little while, what two molecules will actually diffuse through this membrane? So have you figured it out? Can you see which two will actually fit through? All right. So it's your purple and your white that are fitting through, but those blue molecules are just too big to fit through that membrane. So we can look at this and say, okay, these white circles could represent the iodine that's in the cup, and the purple would be the glucose, and the blue could represent the starch because it's so much bigger than the glucose. Okay, the next thing to look at, we then looked at osmosis. And remember, osmosis is a little different than diffusion because it has the special requirement that it's diffusion of water through a semi-permeable membrane. And we looked at this when we took our red onion cells, we looked at them under the microscope, then we put salt water on them, and then what we were supposed to then put regular water on them again. And you'll see that in this video. So remember, one way that osmosis is defined is the movement of water from a high concentration of water to a low concentration of water. I find it easier to remember osmosis as water moving to wherever there are more solutes. So in our red onion cells, when we put salt water over them, there was more solutes, which was in this case salt, in outside of the plant cells, so the water moved or diffused out of the cells, and that's why the purple cytoplasm shrunk so much. So let's take a look at this happening, and then you'll also be able to see what do the cells look like after you put regular water on them again. This is done rapidly, so they sped this up multiple times. But you can see how small they got, and now here comes the water is osmosing back into the cells, and look, they're going back to looking like normal. All right. Now let's take a look. This should look familiar to you from your lab. We're talking about what happens to cells when they're in different solutions. We can call those solutions hypertonic and hypotonic and isotonic. So we're going to look at, well, what happens in each of those situations? So if you look at this first one, now this is a blood cell, a human blood cell, and this is a diagram of a plant cell. So in this solution, the blue arrows show how the water is moving. You see in both of these cases, the blue arrow is the water is moving into the cell, and here the water is moving into the cell. Well, I know if my water moves to wherever there are more solutes, there must be more solutes in the cell. That means there's less solutes outside of the cell. So my solution that my cells are floating in must not have very many solutes, or they're lower in solutes. So we would say that they are hypotonic. Hypo means low, just like if you have hypothermia, your temperature has gotten too low. And so hypotonic means that it's low in solutes in the solution, so water is going to leave the solution and go into the cells. The bad part about human cells is that we don't have a cell wall. So if too much water goes in, our cells can blow up. And that's not a good thing for our cells. Now, plant cells have a cell wall, so they just get really firm and full of water. Okay, in the next situation, you see I have water both going in and going out, which means there's the same amount of solutes inside the cell as in the solution. So we say that this solution is isotonic to the cells. Now we have our last one, and we see water is leaving the cells. Our poor little cytoplasm has gotten all teeny here, and the membrane is just stuck to the cell walls in a few little places here. And look at our poor blood cell has really shriveled up. So if the water is leaving the cells, it means it's going to wherever there are more solutes. So there are more solutes in this solution, so it's called hypertonic, because there are more solutes in that solution. I hope that helps you get your homework done, and you can always go ahead and send me an email if you still have some questions about this.